So can you start by telling us your name, your institution, and your role?
I'm Ross Wilson. I work at the University of California, Berkeley, and at the Innovative Genomics Institute, where I am serving as the Director of Therapeutic Delivery.
In a couple sentences, can you describe your research project?
Our project has three major efforts. We overall are using CRISPR to remove pathogenic regions of the genome that can cause various diseases. So one of the major indications that we're focused on is Huntington's disease, and the other one is a different expansion repeat disorder that can cause ALS or FTD-like symptoms. And we're using viral and non-viral strategies to deliver the CRISPR enzymes into the central nervous system to silence these pathogenic expansion repeats in the genome.
Great. And then within that, what specifically do you work on?
My lab is largely focused on non-viral delivery technology and developing new strategies for delivering CRISPR enzymes into the CNS and other parts of the body. And my lab has actually expanded our efforts into improving viral delivery technology as well in support of our SCGE project.
What is your favorite thing about the research that you do?
The thing that I really love about our research is being able to collaborate with other people from different backgrounds. It's so exciting and challenging and stimulating to work with people who have totally different backgrounds. I came from sort of a biochemistry background, which is a little bit unusual in the genetic therapy space. And it's been so exciting to connect with neurosurgeons, for example, who bring a totally different perspective to the problem solving that's necessary in tackling some of these really challenging, novel, but valuable strategies to bring genetic medicines to the clinic.
So what is something cool or interesting that you found in your research?
One thing that's been really interesting is looking at the ways that delivery impacts editing outcomes. So in the field, usually people think about CRISPR as this pair of scissors that just goes into the genome and gets the job done. And that is largely true, but it can make a huge difference how that enzyme reaches the nucleus of the cell that it's trying to edit. So my lab has largely worked with preformed enzymes, which kind of go into the cell as a ready-to-go active complex, let's say. And those enzymes are really good at getting the job done because they're instantly fully functional. In contrast, when people use viruses to deliver genome editing enzymes, there's a really complicated process between the virus delivering genetic information into the nucleus and that genetic information sort of maturing and transforming into the signal that generates the CRISPR enzyme inside the cell. So it's a much more convoluted pathway, and looking through the literature and the history of viral CRISPR efforts, it seems that there's some, let's say, drawbacks or complications or shortcomings that have made viral delivery of CRISPR maybe more complicated than people initially realized. So it's been kind of exciting to revisit this question and maybe find new ways to use viruses to deliver CRISPR enzymes, hopefully improving outcomes and potentially supporting more successful clinical trials.
What is an advancement that you think will happen in genome editing in the next 10 years?
My hope for the next 10 years is that CRISPR therapy is going to become more generally trusted and understood in terms of its safety profile. The reason that this is so exciting is because once we kind of know the rules of genome editing and we understand what is likely to be safe versus what is likely to be risky, it's going to allow the inherent convenience of CRISPR editing to transform into broad clinical impact for diseases that would otherwise be left behind. So to put this another way, the reason CRISPR is so valuable is because in the lab, it's very, very straightforward to reprogram the enzyme to go after a different region of the genome. theoretically, the same thing should be true in the clinic, but we can't do that at full speed, so to speak, just because we don't totally understand the safety profile and all of the risks. So once we get better at understanding the risks, and once we build more and more trust through early clinical trials, people are going to be able to use CRISPR in the way that makes the most sense to sort of redirect it to multiple genetic targets. And this will help address lots of patients who have rare or even unique mutations that would otherwise be left behind because it's not profitable to go after this sort of N of one therapy. So that's my hope for CRISPR, is it's going to really truly democratize genetic medicine.
Changing topics a little bit, how do you engage with patients and patient advocacy groups in your research?
Yeah, so working at the Innovative Genomics Institute, we've had a long-standing effort to use CRISPR to address sickle cell disease, and that has been a huge opportunity for me to be exposed to sickle cell disease patients and advocacy groups. It's been really valuable to get those perspectives from people sort of on the other side of the spectrum. We're in the lab really at the earliest stages of developing medicines, and it has been really interesting and moving, frankly, to interact with people that could potentially consider benefiting from these therapies.
In that same regard, how do you consider health equity in your research?
Health equity and accessibility of genetic medicines is something that I've been thinking about very seriously for years. So working on delivery technology, it's sort of inherent that if you can find better and more affordable and safer and more accessible ways to deliver these therapies, it will instantly allow access to become broader. For example, the first FDA-approved CRISPR drug involves a really labor-intensive, risky, and complicated cell transplant. And if we or other people working on this challenge are able to find ways to bring CRISPR to the cells inside the body instead of doing a transplant, this could totally transform the total impact of these medicines. So that's one way that technology can directly change access. Another thing that we think about a lot at the Innovative Genomics Institute is all of the non-technical contributors to the question of access and affordability. So even when a cellular therapy gets transformed into an in vivo therapy, it's very likely that the companies that deploy this will continue charging the same high price for that product. So we think a lot about why those prices are so high and what sort of structural things could be addressed to change those incentive structures, so to speak. We really want genome editing to be broadly accessible, and at the moment it seems to be extremely expensive, and we've put a lot of work into this. One thing that we did at the Innovative Genomics Institute is assemble a affordability and accessibility task force, and we just published a paper that reports all of our learnings from that one-year experience. So this was a really rewarding thing that I was part of.
Great, thank you. And then is there anything else that you would like to say about your work with the SCG or your research related to that?
Working with the SCGE has been really like a defining part of my career. My lab has been part of the SCGE since basically the earliest days, and the way that it brings people together and kind of removes barriers is so valuable. It's been wonderful to talk to all the other scientists who are facing the same challenges that we are, and it's really injected a lot of excitement into the CRISPR delivery field, which is something that everybody says they want to be a priority, but a lot of, let's say, funding agencies have not been bold enough to support novel delivery technologies. So the SCG has been a real leader in sort of putting their money where everyone's mouth is in terms of supporting these delivery efforts, because they will have a huge impact in changing the landscape for who can benefit from genetic medicines relying on CRISPR or even other types of strategy. So yeah, it's a really bold initiative, and it's been so exciting to be part of it.
Okay, thank you very much, Dr. Wilson.
Thanks.